Hello, everybody. I'm Dr. Amrit Kashyap, and I am going to give a presentation on constructing an ODE model for fMRI data. So fMRI data is thought to be the sum of both sort of network activity as well as task-dependent activity. And so this network component sort of comes from these cortical loops that exist already, and we are hoping to construct an ODE model to represent this network activity, and then we're going to validate that we have this network activity because it looks like a biophysical model of the brain, and then we're going to try to decode the stimulus-dependent activity by removing the network activity. So why does this matter? Why do we try to separate it? Well, we're trying to understand brain signals and what fMRI represents. And here we have a sort of graphical summary of our work where we take sort of a task and we split it into the network activity and the stimulant dependent activity. And then for each of the task behaviors, we have the reaction of how long they took to perform this task. And then we use this as a sort of a gold standard metric to correlate with the actual neural activity. And we show that our separated activity has a much higher correlation than our original. So a little background on how neural simulations have sort of evolved. So at the very bottom here, we have neurons in this abstraction pyramid. At the very top, we have sort of the entire cortex. And at the very bottom, we have had really good measurements and intracellular recordings. So we have been able to construct almost perfect equations to represent the activity, including the very famous Hodgson-Huxley equation. And later on, we have higher up in the pyramid, it's slightly less clear exactly how to model it. And so a sort of a rule that has been used is by taking sort of individual elements of the lower level, and then you sort of sum across a layer structure, and that will give you sort of a model for this higher representation. So in the grand scheme of things for fMRI modeling, where you have sort of whole brain scope and very slow activity, the two main camps that are sort of popular these days is one is the neural field equation that takes the T1 image and estimates the surface of the cortex and then estimates the eigenvalues in the surface, and then sort of uses a linear combination of them to simulate fMRI signal. The other big famous model here is the brain network modeling, which uses sort of white matter tracks, and that's how they sort of construct a network of which brain regions are connected, and then they model sort of individual brain regions with sort of a complicated neural mass equations for the lower levels, and then they sort of aggregate together across the network to give their activity. But in this in our goal to separate this activity, it's not very clear from either of these models as how this local activity that's dependent on the task is sort of within the model. So instead of these two approaches, we are using much more of a DCM approach, where we sort of learn the connectivity and learn the equation directly from the data. And we're using a sort of the next a sort of more recent version of DCM called Cindy, which is scales much better than DCM, so you're able to construct a whole brain model. And the way that Cindy works is that it basically constructs a massive regression problem to represent the derivative as a function of the variables in there. And the way that it does that is it creates a library of functions. Since x, y, and z here for Lorentz system are recorded, you have x, y, and z. Then you can also square it and then multiply it and whatever. So you can create a huge library. And the problem here is that so you have all the derivatives over time, which you can compute by the difference, and then this massive library. And so you solve for these little coefficients, and you want these sparse coefficients. So right here, you have the Lorentz system, and it solves for exactly the exact coefficients that it was simulated for. The data that we use a whole brain model using the desc and Kiliani atlas with 84 both cortical and subcortical regions, and we parcellate it based on the individual space, not on the MNI group space. So the segmentation is done on each individual, and then we sort of average the signal in these brain regions, and we do very minimal pre-processing, just a bandpass filter, averaging, and normalization. And we also get the we've also done the tractography for 443 subjects to estimate what the connecting was like, in order to validate our model is biophysically plausible. So how do we validate this? Well, when you are sort of constructing a model from the data, first of all, you have to be a little bit careful, because you have to make sure that the model that we're getting is not something nonsensical, but it sort of matches the structural connectivity that we get. So here, I'm calling this the effective connectivity, the different orders of the Taylor series coefficients. So first order, second order, and all the way to the nth order. And we're going to look at how all of these coefficients are related to the structural connectivity and make sure that it represents a biophysical model. The second thing that we're going to do is that, I mean, we train the model on predicting the derivative. It better be able to predict the derivative. So we have to look whether or not it can predict the derivative. And the third thing we're going to use to validate the system is that we're going to put an impulse response and make sure that we have a hemodynamic response function, because this is going to be important in decoding the signal later on. So the coefficients, if you look at the coefficients that we get, so this is each one is for the different higher orders. So this is the first order, second, third, fourth, fifth, sixth, and seventh. And I've put all the odd order coefficients on top and all the even on the bottom. And you can see that the odd order coefficients have very similar structure to the actual measured structural connectivity, especially in the intra-hemisphere connections between within each hemisphere. And they have pretty high correlations, over 0.4. And so how do we sort of make sense of this giant system? Well, we can also see that the coefficients for the even orders are much, much smaller than for the odd orders. So the vertical bar here is these two have the same magnitude, and these two have the same magnitude, these two. And you can imagine that x cubed is much, much, much smaller than x squared. So these coefficients being that large are much more relevant. So this looks like an odd function. So we basically took the structural connectivity matrix and we saw how much it goes into each one of these. And we constructed an estimated one-dimensional function, what it could be. And you can see that in the red line that it looks like a sigmoidal function, and it lies between other fairly well-known odd functions that are used in the brain network modeling community to simulate. So without any assumptions, we basically get something that looks like a brain network model with some differences that I'm not going to get into, but in basic sense is a structural connectivity in the argument of a sigmoidal function. So how does it do on predictions? Well, here's like a sample observed derivative for a single resting state scan of 15 minutes. And you can see that the predicted derivative has a lot of the same features as the observed derivative. If we look at this per brain region, we can see that most brain regions have very high correlation, except for those in the very frontal regions and then temporal regions. Those don't do so well. And this is because we think that the signal in these areas in fMRI are generally poor, and this also includes sort of subcortical regions, although the thalamus and the cerebellum do pretty well in this model as well. And this equation, we train it on 400 individuals, and we test it on 50 other individuals, and we can see that a single equation is really able to capture most of the dynamics of the system. And it does much more poorly for task than for rest, because we were trained it on rest data here, but more on that sort of in decoding sections later. We can also take the sort of the initial condition and integrate it over time, which gets this sort of integral, and which we can compare directly with the resting state signal over time. And it does a little bit more worse because the integral is dependent on the initial conditions. So further away from the initial conditions, it gets worse and worse. But it still explains a pretty good variance. I sort of had simplified a lot of these things, but basically there is a trade-off between getting all of the dynamics versus getting all of the structural connectivity, and that is because when we have sort of a sparsity parameter of how many coefficients we want to represent the system, when we have very few coefficients, then we have very high correlations to structural connectivity, but then we don't capture the dynamics. And when we have a lot of coefficients, we can capture the dynamics very well, but then it starts looking less and less like a biologically possible model. So we wanted to have some sort of trade-off here and sort of try to pick models that can do best of both worlds. And we also looked at sort of the decay rate, which here is the sort of the diagonal terms, and we assume the decay rate as a part of our methodology because if you look at it, at the sort of the diagonal terms, you have the decay as well as the local input coming in the noise. So the algorithm is not able to separate these. But if we assume the decay, then it sort of solves for the rest of it and then puts the rest as noise. So it's important to assume this sort of decay rate. And the decay rate, you can imagine, is when there's no more energy in the system, it sort of just exponentially decays. So you can fit it with the SBM function. The value of the SBM decay rate is around 0.22. And you can see that our lambda of negative 0.21 does pretty decently out here. So that is the model that we sort of used as best capturing rest for now. And also the HRF sort of varies between regions, but so we picked sort of the average HRF response here, which is what you plotted. Code. Once we have this model, how do we decode our data? So our what we do is we estimate sort of the processes that are in rest but not in task. And the way that we do this, I will get to it a little bit later, but let's assume that you have all the processes that are in rest but not in task. And once you have those, you can just directly subtract it from the signal. So here we are looking at the derivative space of a single brain area, and with respect to all the stimulations. If you align all the stims with sort of you align them when they all occur, and you take a sort of average response, you start getting this sort of HRF looking response. And you can see that the measured response has a much smaller HRF than once we separate it. You get this sort of boost. Now, does this mean that this increased HRF actually correlates to our behavior variables, or are we just sort of increasing the signal? Well, if you then correlate it with the reaction times, so how long they took for each of these purple tasks, you do see a bump in correlation for both the magnitude signal and as well as the original derivative space signal. And this occurs not just for the single brain region, but sort of across all brain regions. You can see sort of this is this separated one versus the original one. You can just see that there's just a lot more color indicating higher correlates. So how does this translate? Well, if you look at the correlates across the brain, you get sort of the areas that are highly correlated are sort of known in the working memory tasks. So the prefrontal areas, the parietal areas, and this matches well with previous publications. But you can also then sort of summarize this entire spatial temporal information. And we used another elastic net, since we've been using regression the entire time, use another regression technique, which basically took a linear combination of the spatial temporal signal and fitted it to predict the reaction time. And here you can see that our separated signal has much higher it explains a lot more of the variance seen in the reaction time than the original signal, and this generalizes across all subtasks. And okay, so I promised that we would get to understanding, well, how do we get the processes that are in rest but not in task? And to do this, we have assumed this sort of active cortex model, which assumes that task is a subset of rest. So during rest, all processes are active, everything is active, and during task, only a certain subset of them are utilized to perform a certain behavioral task. And to get the processes that are unique to task, that are unique to the task and not in part of REST. We basically take the REST model that has all processes and remove the processes that are unique to task we train a model that is unique to task, we remove it from the REST, and then we get all the processes that are in REST but not in task. And we did a sort of a generalized sweep of this by having sort of coefficients C1 and C2. And here we can see that all the diagonal elements when C1 equal to negative C2 fit the system best. So basically this equation is showing that you take the measured signal and we're going to remove the processes that are background processes that are only in REST but not in task. And we get these processes by taking our model from Cindy that encapsulate REST, and we subtract the model trained by task to get the difference of the processes, and then we remove those processes and we get our reaction time. So a comparison to what we just did here is that, as you can remember, is that previous models sort of just had to simulate these complicated network systems, and so they had metrics that they had to use on top of it, such as like functional connectivity, in order to compare between the simulated models and the actual activity. We've moved to directly predicting the derivative and the observed derivative, and this allows us to then separate out what is the observed part, what is the network part, and what is the task dependent part, and gets really good fits to the behavioral metrics. A similar method using DCM has fitted at an R of 0.46, which would be an R squared around 0.2. And here we have an R squared around 0.6. So this is a substantial increase, mostly due probably to the fact that our system scales much bigger. So we're able to get the full Taylor series expansion of the system with all brain areas, and we're able to then decode. In conclusion, we've shown that we can estimate an ODE for the measured fMRI that captures the network activity for that data. We've validated it by showing that the function generalizes to the individuals, not to just a training set, and that the network function is very similar to what has been used to simulate other models. And it's a nonlinear sigmoidal function with an argument having the structural connectivity matrix, and the impulse response looks like the hemodynamic response function. And when we have this active cortex model where task is a subset of REST, we can also then decode the signal. Thank you for listening. I would like to acknowledge all my team members here from the Virtual Rainbow Lab here in Berlin, as well as people that I've worked with from my PhD days, my Atlanta collaborators, and thank you all for listening.